Hello. Good morning, sir.
Good morning.
Yeah. So looking forward for the session. Last what we discussed was based upon the p-value, how the p-values are calculated, what do they actually mean, what how the conclusions can be made based upon the p-value. Yeah. And later on the second session, we discussed more about the statistical test, which tests are used and how the conclusions can be made by performing those statistical tests. And yeah, we also generate the p-values from those tests, and we saw that as well. In this very third session, what we are going to more deep down is about the test on categorical data. The test what we saw in last session was on continuous data sets. Now it is more about uh, categorical data. So
Perfect.
we'll be
Thanks.
going forward with this. Thank you, sir.
Thanks, Akash. So please be interactive and please raise your points, queries during presentation. See, as we see that last two times that with this continuous data, how different statistical tests would be there. Now we are going to have some proportion agreement and rates. So basically, it looks quite easy to calculate. Like in our general and routine day-to-day life, many times even in day we keep on calculating proportions. How many times, how many number would be there. But while you talk about the data point, yes, while you talk about the data point, then it's become little tricky to. Could you mute if somebody is having accidentally being raising any point? Yeah. So where it's coming sound. Okay. So what happened? Yeah. So then it's quite easy to routinely whenever you talk in our day we commonly calculate proportions, but while we talk about from the data itself, it become little difficult or tedious to calculate these proportions from there itself. And let's go with that. Ankush, could you have a look on that? Is it possible to mute if somebody accidentally put something over there?
Yes, sir.
Now done. Okay, thanks. I hope you can see my PDF file, right? Could you see my PDF?
Yes, sir. Yes, sir.
Okay, perfect. So let's say that there we will talk about a couple of six statistical tests today. And before statistical test, we will see how we can prepare our data. It's a very simple way to prepare our data. We will look upon how to make that Excel file to have how to make your Excel file such a way that you can perform these different statistical tests and prepare data, and simultaneously we will also have the dashboard. Point is that many times many articles only ask for these simple statistics. It's not very advanced statistics. These are not very complex statistics. Only you need to have your data managed properly, and that managing is itself is not difficult. Only you have to know how to manage your data, how to prepare your data before performing these statistical test. And these are very powerful tests. By looking these, you can understand your data. Reviewer, your mentor can understand the gravity of on a plan of your data. Everything comes with the very sweet and easy manner, having these proportion and agreement associations on stratified rates. So as you know that proportion or risks, this terminology, be very specific on that. Whenever you are writing your manuscript, writing your report, don't get don't loosely use this word, because they are having calculating proportion or calculating risks. We will go with next slide. they are little different. However, both of their calculations comes like same way, like having simple calculation x upon n, and this x is number of event occur, and out of how many people are having occurred. So these are these basically, proportions or the risk calculations. But see here also the event is basically event given by percent time. So these many times on odds is basically event versus non-event. So this terminology many times we use very loosely. We think all are same, but in reality their background, their way of calculations are different, and then it makes quite uh, thin difference when you are going to make the calculations, when you are making the interpretations, when you are talking about take it forward about your results. So be very cautious to calculate or make the terminology for your calculations from your data. Believe me, from your Excel file, from your data, these are not very difficult to calculate, but their interpretations, the way it is make value, that makes quite difference over there. So like here, deeper point is that a proportion answer what fraction, a rate answers how fast, and we will see that how the proportion and odd base pooling a ratio works over there. And there is a one thing is that uh, this time we are having surprise, so we are going to give you one website, one link that very easily you can use it. It's completely free, and from ourselves we prepare for you, so you can be out of SPSS, any proprietary software, freely you can use it and get results. So that is another surprise gift we are having, and we will show for that. So let's say we are having one table we are having here that Lancet Oncology Ginaini et al. Published, and from 107 patients, and they distribute 107 patients per arm. So it was a randomized control trial, and both arms were their control arm and pertuzumab arm, and those patients were treated, equal number of patients were treated with 107 and 107, and out of that PCR yes was for 49 for pertuzumab arm, and no was 58, and then control was having PCR yes was for 31 and followed by 76. Now from this simple table, we will show in Excel file how to prepare the data to having this simple table. Even many of us get challenging how to calculate or how to prepare this simple table. It looks quite simple, but you have to have your paper data such a way that you can make this table. Okay, so once you have this table, from here very easily we can calculate these three terminology, or four terminology like risks, odds, risk ratio, and odds ratio. Okay, these are these four points we can calculate. So, and these risks are basically calculating risks are basically stand upon two parameter like risk 1 and risk 2, if you see. And this risk 1 is stand with how many we are having, for particular, for pertuzumab arm, how many were treated. That was, you can see, 107 were treated, and out of that 49 were having PCR. So that is this risk of having PCR, those who were treated with pertuzumab was 0.458. And then similarly, risks of having PCR, those who are in control arm was 31 upon 107, so 0.29. So apparently we can see this risk is quite high. Means number of people who were having in PCR was quite high in pertuzumab arm as compared to those who are in control arm. This typically called risk. This is not this risk ratio. And we are not talking about the rate here. We are talking about the risk here. Now, once we are having both risks, like risk 1 and risk 2, and then we are going to have the risk ratio, means risk ratio stand with ratio of these two risks. And it's also depend risk ratio of R1 divided by R2. That is 0.458 divided by 0.290, and then it's coming for risk ratio. What is the risk ratio? Risk ratio would be interpreted as that getting PCRES, those were in pertuzumab, is 1.58 times more as compared to those who were treated with control. The risk ratio, that's mean says. So while you are using the term called as compared to control, so here we are saying about the risks of having PCR, those who are treated with pertuzumab. Risks of having control PCR among control is 0.29. But while you are talking about risks of having PCR in comparison to control, mind the word. So risks of having PCR in comparison, risks of having PCR in pertuzumab arm in comparison to control. When you are using in comparison to control, then you are using the terminology called R1 divided by R2, and that is called risk ratio. Similarly comes with odds ratio. Odds is basically how many were in PCR as compared to non-PCR people. See here, we are talking about saying as a O1, that is nothing but 49 divided by 58, that is ES-PCR divided by this 58, and whereas for control arm 31 divided by 36. So getting odds with no means yes divided by no, that is called odds. Risk is basically denominator is having overall people, but here denominator is your fraction of people, those who are not having PCR, and thereafter we are going to calculate odds ratio by O1 divided by O2, and then odds ratio is basically calculated, the same way, 49 multiplied 76, basically these calculation is going to have, and then it's called as a 2.07. So now think that once you are presenting your data, presenting your result, if you use that terminology, people use very reluctantly use this terminology, but in comparison to from the same data, same table, once you talk about odds ratio, once you talk about risk ratio, how much difference is going to have? 1.5 becomes 2.07. Just same data set, the same value. There is nothing wrong in there, no changes in there in terms of the calculation. Only just no changes in there in terms of the values in the cells. Only in terms of this calculations we are having risk ratio, it's coming 1.58, and once odds ratio is coming quite high value. So very cautiously use that terminology, and trying to have the uniform in your manuscript or in your report. Don't make somewhere your risk ratio, somewhere your odds ratio. Don't do that. Let it be uniform across all places over there. Okay. And before going for incidence rates, do you have any points or any concern we can discuss?
So can you explain what is ratio below one concerned as in more than, here it is more than 2.0, so does that mean the risk is out of some bound or something, or is it same as uh, twice or
Just
something?
you
How
explain,
do we
you are
interpret
explaining
this?
I think you've touched a very relevant point. Just explain it more. Yeah.
So I just wanted to know or ask you if the risk here it has shown as 2.07, odds ratio has increased as 2.07, uh, and the odds, it's one is 0.408. No, sorry, that is that is just the ratio, sorry. uh, Risk ratio is 1.58 and risk odds ratio is 2.07. So what my concern was, uh, do we treat the increase in risk as same for, because it is 1.58, I might consider it as 58%, and for it is 2.07. So is it 2% increase in by 7%, or how it is, like, since it is more than one?
I
more
think
than
I
two.
see, I see,
Yeah.
I see, I see, I see, I see, I see. So see, uh, when you are talking about risk ratio or odds ratio, I think you are trying to raise the point about the reference point. So reference, we use this terminology called reference point, and reference point is nothing but it means always call as a one, because one means there is not any difference between two risks. Let's say you are having 0.58 here, and somehow for R2 also we are going to get 0.58, 0.458. So then R1 divided by R2 is going to be exactly 1. Okay, so 1 is always considered as reference, whatever it is. Is it a risk ratio or odds ratio? Similarly, if you are having the same number of PCR happen and not happen for O1 and again for O2, then definitely you are going to have odds ratios also as 1. So whatever you are going to get, risk ratio, odds ratio, 1 is always the reference group. Now what might happen? Either your risk ratio could be below 1, exactly 1, or above 1. So for example, here we are having above 1. It could be below 1, it could be equal 1. Similarly, odds ratio could also have below 1, exactly 1, above 1. None of them could be below 0. So minimum value could be at the most 0. It can't be negative. So it should be between 0 to 1, exactly 1, 1 and above. So if it is a exactly 1, so it will say there is not any difference in terms of the risk ratio. If it's odds ratio coming 1, no difference in terms of the odds ratio. If it is a below 1 for both risk ratio or odds ratio, then we are going to say it is a that certain percentage low of risk ratio, that certain percentage of low with odds ratio. But here in this example, as we stick with this, here it is having 0.58% risk is high in pertuzumab to getting PCR. 2 times more, 2.0 times more. So here we are talking about times. So 2 times more to having PCR, those who are in treated with pertuzumab arm as compared to those who are not treated with control arm. Does it make sense now?
Yes, hello, sir. I have a small doubt.
Yeah.
Sir, here we are using pertuzumab. So, and PCR, if it is yes, it is considered as risk. I would like to know why, because the drug is working here and the tumor and everything is now complete response. So, it should not be a risk, right? The patient is non-risk now.
Oh, I think you are talking about in terms of this, trying to link with this clinical relevance on that PCR, yes or no. No, just to avoid it, because we're using, it is the results obtained from that, and from there itself we are trying to make the interpretations. Maybe we can avoid to have the clinical linking rationality why PCR coming with pertuzumab arm more than control arm. That logic may be avoided, and it's a by chance, basically. It could be other way around, but the calculation is going to be same,
But sir,
right?
I guess it is not compulsory that always the outcome is bad, right? Here the outcome is PCR, which is good thing, and the as per the risk ratio might mean like risk ratio or odds ratio, but it shows the chances. Like just forget about the risk ratio once. Consider the odds ratio. So odds ratio says like 2.07, which is like the odds of having PCR yes, which is complete response, right? Is higher in pertuzumab arm as compared to the control arm, which goes right. Would it go, sir? Yeah. Is
Yeah, yeah, yeah.
So, though our outcome here is not bad thing, outcome is a positive thing. So that is on us how the study is designed. For here we can say that outcome is good thing, and the chances of having a good outcome is very high in the treatment group as compared to the control group. And I also first got like the risk makes us like think always about risk, because we mostly do the bad
Risk is
outcomes.
bad thing. Risk is bad thing.
Yeah, so one thing what I wanted to like for this particular scenario, which would be the correct outcome to consider, risk ratio or odds ratio? Like
Yeah, that's a very, very relevant point. Yeah. So my answer is that not an uniform one. It could be both. It's depend upon where you stand with your hypothesis. It's depend upon before having trial, before having your data, what you mention in your protocol, what you mention in your synopsis. If you mention in your synopsis that you will go for risk ratio, then go with that. If you mention in your synopsis or proposal that you will go for odds ratio, go for that. There is not in this scenario, in this case particularly, whatever been mentioned having your pre data having, okay, that should be retained over there. Okay, don't jump and say that risk ratio is good for me, so I'm going forward with that rather than odds ratio. Don't go for that. Rather than stick what you committed that you will do in your analysis. That's why statistical analysis plan stand very relevant, and that then take it forward. So whatever mentioned in your statistical analysis plan, that should be remain over there.
Understood, sir. Yeah.
Okay, so then again we will go for another terminology that is called incidence rate. And as its rate says that rate, so rates always comes with some with respect to function of what's coming this thing. So this is coming by calculating with respect to some percent time or with respect to some time. And then it comes as incidence rate, like event divided by total percent time. Like quite often we use like our clients et al.'s paper with BMC palliative care. We are having two arms called arm A and arm B, and adverse event calculated for both arms with 18 and 30. And the patients were observed for 120 months and for arm A, and patients were observed for 110 months for arm B. And then we are trying to see that event rate like per month. So 18 divided by 120 and 30 divided by 110. That's where we are calculating incidence rate over there. Okay. So it comes as per patient's months over there. And then if you see that raw count, if you see the raw count, obviously here it is coming 18 versus 30, or only fair because follow-up is similar. But if A were followed twice as long, it count could still mean a lower rate. And that similarly from incidence rate, we can also calculate prevalence. And the prevalence a proportion who has its now versus incidence a rate, how fast new case arise, and that different denominator, different questions comes out of that. So again, you have to mention predefine in your analysis plan, in your proposal, that you are going to calculate incidence rate, and that's the way it should have calculated. Okay. So if you see, there is a one paper called IMG therapy for infractory pheochromatoma and paraganglioma, and open label single arm phase 2 trial. And they were having test one observed proportions against a fixed benchmark value. A standard single arm phase analysis been given, and the exact version should the small samples for early phase trials. So, and they in that questions in that published article, they say that 4 responded out of 16 patients, so 25% beats the 5% benchmark. But with only 16 patients, could 4 response arise by chance? If true rate were really 5%, the exact binomial test answered exactly that. That probability was 0.007. So chance is unlikely explanations. It's actively single in real. So under this null expected number of responder, it is well below 1. So normal approximations was unreliable here. And then we go for binomial trial test. We're going for, now simple example. Let's say how we are going for binomial simple example. So once we are having data, let's say it's your Excel file, and and in Excel file, sorry. you put your patient number 1 to 16, like 1, 2, 3, 4, 5, 6, 7, 8 to 16, and the responses are given as 0 or 1. So if they are responding, so we put it as 0. Not responding, then we put it as 0. If they are responding, then we put it as 1. And same way we put for all the 16 patients, and from there onwards we calculated how many were responder, that is 4. So 1, 2, 3, 4. This is the simple example file we need to create. Excel file we need to create over there. There are some statistical tests, like you can go with Excel, you can use this formula in Excel itself, you can calculate, but we are going to have some other way now. We are going to demonstrate how we can calculate this proportion, one proportion test. The background is that we are going to see what is the exact proportion is going to have. So let's see with the dashboard. Could you see my dashboard now? Anyone can confirm?
No, sir.
No?
No.
Gosh,
Okay, let me share again. Could you see now?
Yeah.
Yes.
Yeah, perfect. So this is publicly available. You can check this link and you can open at your place. So let's say for one proportion test, simple one proportion test. So there is in this dashboard, you are having two options. You can upload your Excel file, like browse, and then browse with that. You can upload your Excel file, or you can save your Excel file as CSV and upload it. And definitely you are going to get this result. But other than that, this dashboard, you can also edit results here, response here by from your own data sets, and definitely you can get the result. So let me go through that. So this is this tab, one proportion tab is available, and this exactly going to do that binomial test value. There is a options called greater and lower. So right now we are having this patient for, as we explained in our slide, that 16 patients, like first 6 patients, then again rest 6 patients, and then finally all 4 patients. So, and their responses are given as 0 and 1. So as we see that there are 4 responders who are having 1, or rest of they are having 0. So see the same things being prepared here. And now if you click on this run analysis and benchmark, you can define your benchmark, whatever the way you want to have. You can have 0.01, 0.05, or 0.1. Depends on what benchmark or p-value you want to retain over there. And similarly, you can have your confidence interval. So if you can, confidence interval could be like changes over there. So we are going to have the conventional, this 95% confidence interval, and then run for analysis. Now, if you click on the run for analysis, it will be definitely, it will show that the observed rate are 25%. That is obtained that 4 were responder, 4 out of 16 people. One-sided p-value coming 0.07 with Pearson's percentage of changes, from 6.8 to 53.85%. So it's varying from 25%. So minimum range is coming from 6.8, maximum is coming out of 50 up to 53.5%, and then expected under null hypothesis comes 0.8 value. So this is the distribution also we can see of these responder and non-responders variations. Now, as I said, that you can update this value itself here. So let's say if you replace this 0 to 1, so now if you run this analysis, now look, it's come down from 4 to 3. Your observed rate has dropped down. Your one-sided test dropped down from shoot up from 0.007 to 0.04. Your percentage variation is also shifted from 5.8 to 43.5 to 3.7. So it's dropping now. And then it says that 18.8% are having responder against the benchmark with 5% that one-sided with 0.5. So that's dependent. And now we can also shift your benchmark from insist of benchmark over there, and then again rerun this analysis. So values are not going to be changed here, but this statistical test, because since you are changing your benchmark, your p-values going to be remain change over there. Okay, so this is the first point we like to explain here. Please feel free to use this app with calculating one proportion test. But one thing that one you need to keep on mind that how we are going to have your data. If you're, as far as your data is only the 16, then with this setup you can definitely use it. Otherwise, Excel file you can upload here. I'm not going to show, but definitely you can upload it here, and then you can run this analysis and can get results. Any remarks, any points now?
What is benchmark? Someone is asking.
Okay. What is benchmark? Okay. So see, we are having some arbitrarily, where benchmark is basically deciding an arbitrary value that the level of acceptance region. So I could say the level of acceptance region. So benchmark is an input considered as an arbitrary value, and based upon benchmark, we are going to get this p-value. So if you decline your benchmark value, your p-value is going to be dropped down. Okay, so basically benchmark is nothing but, you can think like it's a passing mark. Okay, so in any exam you are having 100 marks exam, but sometimes we consider 30% is your passing marks. Sometimes you consider 50% is your passing marks. Sometimes you say 60% is your passing marks. Benchmark is something like that. It's basically your passing marks you are going to decide, and you will decide arbitrarily. Before data analysis, you are deciding my exam's passing mark should be 40% or 30%. That's all about the benchmark is going to have. Based upon that, how many you are passing, that is decided by the p-value. Okay, does it make sense on that?
So can it be considered as the reference proportion for alternate hypothesis? Like,
yes, definitely.
Connect it.
Yeah. Okay, so again I have to go and again share that. Any other question?
So how can we decide the benchmark, which value to consider? One question.
Yeah, so generally by default we consider 0.05. That's where we are going to present it. Commonly people adopt benchmark as a 0.05, but based upon your results, based upon your situations, you can decide, but don't go more than 10, 0.1. And generally we go stand between 0.01 to 0.1. So you can go from here, from here to here. That's the maximum range would be having, but commonly we go for 0.05 here.
It's the interface created by sir itself. We'll share the link in the mail
Or
post session.
yeah, we can say share in the mail itself. Okay, and let's say again go for I have to again go to my presentations, unfortunately. So now you can see presentations. Let's come with the presentation again.
Yeah. Yes, sir.
So this is where we are standing. Yeah.
Yes.
Any more points, comments?
No, sir.
Okay, perfect. So this is the one example with this published article where statistical issue and recommendation for non-inferiority trial, and we are going from 1 proportion test to 2 proportion test. Okay, so where we are standing on the 2 proportion, so we are saying no odds than. So that's very important. Then this article also says that difference in proportion versus pre-specified margin, that is delta. So until now, we say that proportion is going to compare only as a single proportion, like whatever is going to come, we're going to accept it. But while we are having 2 groups, then, or predefined groups, or predefined proportions, then we are having a 2 proportions comparison. Okay, That's the area coming the two proportions comparison, and that's why we need to specify the margin of difference. Like like two party in election stand with that. So obviously one party win over another party, but how margin they are winning? Is it a 5% margin of the total vote or 20% margin of the total vote? That is basically nothing but the delta value here. And if it is a broader, so that means the big difference is there. If we are narrower, then big difference is not there. Obviously, in the political, voting, election case, whatever which one is winning, we are saying that definitely they are win party as compared to another. But in medical line, in a clinical scenario, there are some other hinges over there. So basically, even we are one arm is better than another arm, but still within this margin, still we consider both arm are same or both arm are not very big different, because there are some other aspect in your life. Because when you are talking about this outcome, let's say we are saying that if you use this 10,000 per pill drug, the success is going to be 50%. Instead of that 10,000 per pill drug, if you use 500 rupees per pill drug, so per pill of that drug would be 500 rupees and another would be 10,000 rupees. But if you use 500 rupees per pill drug, or 10 one strip would be, let's say, 5,000, whereas another drug would be having, one strip would be having 1 lakh rupees. Now if you use 1 lakh rupees drug, success rate is 50%, whereas you are using 5,000 rupees per strip drug, your success would be 45%. Obviously, that 45% is lesser than 50% success rate, but there is a negotiation because it's a huge amount. Are you going to really invest that amount to getting 50% success rate or not? Maybe some, it's not about this percentage or success rate in terms of the life or death. It could be simple control these, viral fever. So person could be controlled viral fever within 2 days, or sometimes it can go for 3 days. So, but with this margin, so it's always having, negotiation that how much impact or how much cost we can involve to getting this margin. So in our clinical scenario, that margin makes sensible, because with many aspects of life we do consider. It's not that only high percentage or low percentage. Even it is a low percentage, still we need to consider some other aspect of life, and that's why the non-inferiority comes over there. So that's why you are going to give the relevancy of the non-inferiority, that non-inferiority asks it is odds by what percentage, because still it is acceptable. Okay, and that's where we are coming out of that.
So
Yeah.
So does that means that we are reducing, so the new drug has definitely reduced the cost of buying it. Thus, it provides a lesser effect or response than that of the traditional or standard drug already been there, but it is very costly to afford.
Yes.
And so does that also drives the research? That's why the non-inferiority studies have been done mostly.
Exactly. Is there any point or questions over there? I can see that there is I think, if I'm not mistaken.
Sir, somebody is asking if you can explain the concept of delta in this sense.
Concept of delta in this aspect. Okay. So see, delta, it's a value from varying value between two proportions. Let's say in one, what we are trying to say, we're trying to say that one arm is having 50% risks or success rate. Let's say one arm is having 50% success rate, another arm is having 45% success rate, and that delta comes 50% minus 45%, that is 5%. So delta here you are going to have the 5% delta. Why you are saying this delta value? Because we know that second arm is going to success with 40%, 45%. First arm was having 50%. So it is a poorer than 50%. It is a 5% poorer. That is the delta value is that 5%. But still, even 5% lower are still acceptable because that second arm is having other impact in terms of the cost, in terms of the low toxicity, in terms of the affordability, everything is good. So even the 5% is varying lower for second arm, still you are going to consider second arm. Okay, and that's where the delta stand for. Calculating delta, there is no way to calculate delta. It's up to us how much or how far we are going to consider the delta value. Somebody can say in clinical relevancy, somebody can say 10%, somebody can say 20%, somebody can say 5%, but it's depend upon scenario to scenario. Sometimes it's becomes quite crucial. We can't even we can't even compromise below 1%. Sometimes it's a very we can consider lower value as well. So scenario to scenario, it depends how we are going to decide this delta value. Does it make sense? Yeah, okay. So here we are going to come how we are going to calculate this non-inferiority with example. So same way, we are having Excel file, and from Excel file you gave, like we have seen this first Excel file, that everybody was responded and non-responded. Similarly, you are having the new arm with 120 responded, 72 non-responded out of total 200 patients. And the same way, 200 patients were given with the standard arm, but out of that 132 were responder and 68 were non-responder. If you calculate in terms of the rate, 72 out of 128 out of 200, it is going to be rate as 64%. 132 out of 200, it is going to come 66%, and the rate difference is coming 62 minus 66, 2% with confidence interval 11.4%. So, and then this is basically, as we say, that margin or delta value is going to be 12.5, and then lower value, lower boundary is coming 11.4%, and then we say that non-inferiority made or just justified over there. So deeper point is that with 50 with arm insist or 200, the same 2% difference gives 21% of non-inferiority fails, and there are some statistical tool you can adopt it and check it how to get this non-inferiority margin. So the nutshell is that the point is that even this percentage looks different, but with this non-inferiority margin, we can certainly say they are not different because we fixed that margin earlier. That is a nutshell. So let's talk about this again example over there. So how can we go? I'm not sure. Again, you can see my slide, my dashboard or not. If not, I have to again.
We can see your PPT.
Share it. Yeah. So here you go with that, with the non-inferiority, having this tab with the non-inferiority. Again, having these options called upload your data, or here we are already having the data. Now just responder are 128 and standard arm we're having 132. Arm are new and standard, and then you are having non-standard, non-responder like 172. So see, 122 and 72, these are total with 200, and 132 plus 68, again it's totaling with 200. And then we are running analysis here, and once you run analysis here, it's showing that difference is minus 0.2, and then decision is that in a non-inferiority margin made. So insist of let's say if you drop our non-inferiority margin, let's say for 10, and run analysis where it says, so see, confidence intervals is coming 11.3, but here it's saying non-inferiority margin not made. So non-inferiority margin we decided earlier before having the data, before collecting the data, we say that our delta value is going to be 10%. But here it's never met, so it is beyond that difference. So that's why you are saying it's not met. But if once you keep on increase from here, let's say instead of 15, 12.5, we are going to say NI met. Look, the difference is coming, okay? And exactly the benchmark is going to replace from here, you know. Yeah. So from here, it started to dropping over there. Okay. So delta should be mentioned again. The nutshell, delta should be mentioned earlier. Responder and non-responder and need to be calculated over there. Here in this example, we are having exactly standard arm and new arm. We are having exactly the same number, but it could it's not that always both should be having equal number. It could vary. Instead of total 200, it could be 180 and 220 or something like different. But still, we are going to get these results, and that's absolutely fine. And this is going to give us the consideration that we are going to consider that non-inferiority margin met or not. What is the ultimate? Can any of you can say that what does it mean that NI not met? How we are going to make the interpretation out of that?
So is it because the lower bound is crossing the margin value?
Yes.
It's this because of what we say that हमने जो डिसाइड किया था कि वो मार्जिन के नीचे नहीं जाएंगे, अगर वो नीचे जाते हैं तो इंफीरियर हो जाएंगे. सो सिंस हमारा कॉन्फिडेंस इंटरवल वो मार्जिन के नीचे चले गया, then we are not hence we cannot say कि हमने नॉन इंफीरियरिटी अचीव किया है. Is it like that?
No, repeat again.
हमने एक मार्जिन डिसाइड किया था.
मार्जिन हम लोगों ने एक डिसाइड करके रखा था.
हाँ,
मान लो कि 11% का डिफरेंस 11% का डिफरेंस हम लोगों ने मार्जिन
पकड़ा है.
पकड़ के रखा है. ठीक है.
हाँ,
नाउ व्हाट्स वी आर गोइंग हियर 11.3 is going to come it here. See here.
हाँ,
Mention here 11. Now we are going to
minus
11.3. What does it mean? What does it mean here? 11.3 means what is it? Stand here.
सो जो मेरा एस्टिमेट आया था डिफरेंस का, उसका जो लोअर बाउंड है 95% लोअर बाउंड, वो मार्जिन क्रॉस कर दिया बाय 0.3%. 11 से ज्यादा हो गया 3%. 11 से 0.3% ज्यादा हो गया. सो इट इज
नहीं. हम लोगों ने बोला था कि you heard that gracing mark in your exam.
Yes.
What the gracing mark? So let's say you already decide the passing marks is going to be 40%. You say, or passing mark, let's say the passing marks is going to be 40%. Now someone got 35 and we are going to give the grace mark. Okay, 35 plus 5 we are going to give the grace mark, and then we are going to say, okay, you pass exam. Even you are getting below 40 cut-off marks, you are having 35, but we add 5 as a grace mark, and then you are passed. But somebody coming instead of 35, 33 as his exam marks, even adding 5 gracing marks, that person is not going to pass. Okay, so 33 is below that margin we already decide. We never say the gracing marks. We are not going to give grace marks somebody 5, somebody 7. We decide that everybody would get gracing marks with exactly 5 or exactly 7. Let's say predefine it. It's a earlier defined that gracing mark would be 5. Now, even the person is having 33. If we add gracing marks 5, the person is not going to reach 40 value. So that person is failed. So similarly here, since we defined our NI margin, non-inferiority margin is 11 as a gracing mark, let's say, then if we are going to get 11.3 as a boundary here, so we are saying that, sorry, boss, you are not meeting this non-inferiority margin, you failed. So there is a big difference between these two groups. Whereas if you increase it, we add the gracing mark more. Instead of 5 that we are having the example for exam passing mark 35, so we are increase our gracing marks. So once you increase the gracing marks, we are saying, okay, you now pass.
So can you give an example with clinical or drug way? Like so.
Okay.
Uh, uh,
So let's say that again, success rate. Let's say instead of gracing marks, let's say 40% is the success rate of your existing drug. Okay, 40%. It's a very standard drug. Do well 40%, but is having impact in toxicity. Means in terms of the controlling disease, it works well, but in terms of that long-term toxicity, it's not good. This standard arm is not good. So we want to bring in market some new drug, new impact interventions that can control that toxicity, because we see that in long run people get impacted with the toxicity, disease toxicity, and they can't tolerate the drug for long run, and they need to they many of them died because of that. So we are okay to compromise from 40% outcome, 40% success rate to 35% success rate with a new drug, because new drug saying that we can't achieve 40% level of success rate as compared to a standard drug that reach 40% success rate, but we can reach up to 35% of success rate, but we are completely free from any toxicity. You don't have any issue. You don't have any appetite loss, you don't have any BMI loss, everything would be well controlled. Are you okay with that? We could not reach up to 40% over the period. Definitely we are trying to attempt to reach the 40% success rate, but with 35% we are saying that nothing would happen. So that 5% we are going to negotiate, we are going to consider the new drug, even it is a lower than existing one, still we are going to consider because it is having other impact. Another impact could be the cost as well, not only the toxicity. It would be very very cheap drug. Why don't you consider that? That's the clinical relevance on that ground. Now, as we said that 5% set as a mark, we can say that, boss, if you come with this efficacy with 30%, definitely you are not going to consider because it is too low. But up to 35% you can come, then we can consider it. Then go ahead with that. Okay.
So up to 35, the drug is, we can say it is non-inferior, not equal to, but non-inferior to the standard drug.
Non-inferior and accepting that region. And below
Okay.
35%, we are going to say, no, we can't consider it. It's a
Yes.
it is lower than that range.
Okay.
Don't accept it.
Yeah.
Okay.
Yes.
So now again, we will come that third point about this Cohen-Kappa agreement beyond chance. So we are going to have this another terminology called agreement analysis, and this agreement analysis is quite often used in diagnostic medicine. Like the same, you have seen that, once you go to doctor having some fracture, you're doubting that there is some fracture in your bone, and sometimes it's been done by CT, and CT could not diagnose that fracture is there or not. Then you go for another MRI. So there are two tests, two diagnostic criteria, and someone saying, yeah, you are having bone fracture, someone saying no fracture, and then you are going for another one, and then saying that yes, there is a fracture and that sort of fracture. So Kappa statistics, agreement analysis, mostly used in diagnostic medicines once you are having two raters, two same thing been detected by two mechanism, two machine or two people. Simple also in clinical relevance also. Somebody saying that you are having tumor by looking your CT scan. Somebody saying no, you don't have the tumor, it's something different by looking MRI. So that could be this scenario, and based upon that we are trying to find and these are typically your percentage value, calculating percentage value, and then how you can go with that. That's where agreement analysis stand. Agreement stand for both treatment diagnostic tool like CT and MRI, they are agreeing with each other or not. If you treat 100 patients, out of 100 patients, your CT saying 60 are having tumor or 60 are having fracture, but same 100 patient was diagnosed with MRI, and then see that no, out of this 100, that CT was saying 60, out of that exactly 60 people, definitely 50 were having tumor or 50 were having fracture, but rest 10 were completely okay. They are not having any issue. However, rest 40, that CT says there is not any issue, but MRI says, look, out of this 40, 5 were having issue, 5 were having bone fracture or tumor. So there is a discordance, there is a discrepancy between these two diagnostic mechanisms. One is saying out of 60, out of 160 are having problem. MRI saying no, out of 100, 50 are having problem. Similarly, CT saying out of 100, 40 don't have problem, but MRI says out of 100, 30 are having problem, not exactly these 40. So there is a discrepancy, there is a disagreement, and how both of them are equal with each other, how where they are agreeing with each other, that's where this agreement analysis stand with that. And then kappa value is one of that, and kappa stand with 0 to 1 value, not 0 to 1 value. If it is a near to 1, that means both diagnostic parameter words come agreeing with each other. Absolutely, they are saying, okay, this 60 is having bone. This similarly, MRI is also saying no, this 60 exactly is having problem. Again, 40 versus 40. Then you are going to have agreement with kappa value is exactly 1. It could be 0, that's mean completely opposite. So it can't be exactly 0, but near to 0. So if it is saying these 60 are having problem, MRI is saying no, these 60 don't have any problem. And whereas this, your MRI CT saying these 40 people are having problem, and MRI saying no, these 40 people don't have exactly any problem. So completely opposite results might happen theoretically. It's never happened, but in theoretically it's possible to happen. That's why kappa value could go near to 0. Otherwise, we go generally we get 80%, 90%. Another relevance of this use this agreement analysis is that while you are going to compare these disease diagnosed by biopsy as well as by CT scan or MRI. So once you go by MRI, you see that this amount of disease are there, number of people are having tumor. But once you go for biopsy, pathologists say something different, somehow different. So how they are clinically, diagnostically different. That's the relevance of use the agreement analysis. So like two readers agreed 85% of scan. So there is a strong agreement. Surely we can go, both are going to have the same results. And some saying that, some of that lack, if the both readers call positive often, and they will agree a lot by chance, how much their chance is having. So that's the kappa value, 0.84 minus 0.5, and then divided by 1 minus 0.05. And then we are going to have 0.68, substantially, but less impressive than raw 84% suggested. Then if you extend this reader by more than 2 readers, more than 2 radiologists, then we call it phase kappa instead of kappa, Cohen's kappa value. And this is the way you need to prepare your data, like positive or negative, like having disease or not, having fracture or not, having tumor or not, or tumor is benign or not, malignant or not. So that's where your all patients need to be classified by first rater, first pointer, first diagnostic tool, either by CT or X-ray, like positive or negative. And secondly, by second reader, second diagnostic tool by CT or MRI, again should be done as a positive or negative. Like here it is saying positive, both readers says positive, but the second patients, both first one says positive, second one says negative. This is where you need to prepare your data. Once you prepare your data, these calculations like P0 and PE comes over there. These are some basic calculations, how we are going to have, how many were having positive, how many were having negative, and how many were having positive, how many were having negative. So both raters say 20 were having positive, both raters say 22 were having negative, but rater A says 5 were positive, but rater B say 5 were negative. Whereas rater A said 3 were positive, rater B says 3 were positive. So that's the 5 and these 3. These are the discordant pair. Rather than that, these 20 and 22, these are the concordant pair. This is called concordant and discordant pair. And out of that, we are going to calculate this kappa value. and you see this is coming as a 68%. They are agreeing with each other, and that's where we are going to calculate this value. Let's say how we can calculate with the dashboard. Yes, so if you go here, Cohen kappa, and then like same way we are having reader A and reader B, positive and negative values given here. Positive and negative value is given here, and then we are going to calculate run analysis. You can definitely change that one and see 20 and 25, observed value with 85%, chance with 50%, and Cohen kappa is going to have 0.6. Let's say if we replace this value somehow, so like here we are all are having positive, both are having positive, but like here, let's see that reader A sets positive, but reader B sets negative. Let's say you also make the reader B as a positive. So just to sake of this example, so if you now look, our kappa was 0.68. Since both rater are agreeing with each other as a positive and positive, if you click on run again, we are going to get our kappa value is going to be rise. So see, it's raised from 0.68 to 0.72. Now let's say we are going to make change here, and then definitely we are going to come with again 0.68. But let's see where we are having again. Let's say make some difference. So let's say positive here, and instead of positive, if you can make negative here, let's see. Even we are dropping now. Okay, so if you keep on having more discordant pair, then our kappa value is going to be completely going down. Otherwise, it will go up. Okay. Any comment? Any points? Okay. So,
sir, can you explain what is PE?
what is P?
E. Chance
Here?
P is 0.5 here, and
yes.
How did we get
so chance agreement, if you see, it is saying as a chance agreement as a 50%. So it is basically the benchmark we are saying. Okay, and again see, it's a benchmark coming as a by default. You can't change it, but it's a by default is benchmark. So below that, we are completely saying they're completely discordant. There is not any agreement at all between these two groups.
Because in your previous slide, I saw there was some calculation for the PE. So
yes, so that is a theoretical calculations comes for PE, definitely. So let me show you. Could you see? So this is the theoretically is coming over here. Yeah. Okay. So this is again with example. You can check with this article, and then Spearman rank correlation. So what happened that we quite often use Pearson, but Spearman rank correlations between two markers also make sensible, because once we are having the skewed data, then we use Spearman rank correlations than Pearson correlations. Why not Pearson? Because Pearson assumes a straight line link and roughly normal data, but in once we are dealing with the small data sets, clinical data sets, then many times we get non-normal data, and then it becomes useful to go with this Spearman rank correlations. And then marker levels are skewed, and the relationship may be curved but still non-directional. And the Spearman handled that, so it's correlate the rank, so it's captured any monotonic trend. It's simply parts Pearson's computed on the rank over there. So here you are having these Spearman Excel files. So again, let's say biomarkers and value are there, and then we are going to see this what extent the sinking percentage is going to have, and then we are going to mark the individuals with different ranking, and thereafter we are going to calculate this value. And again, we will jump with our dashboard. Let's see. So Spearman here, like two biomarkers value are there. We are going to make these biomarker and sinkage correlations over there. Run the analysis. You are going to get that p-value 0.001. Biomarker and it says that positive correlations is there. So what does it mean? What's the basic fundamental relations, hypothesis, and what these p-values stand for? So here you are saying that eight pairs means 8 individuals biomarker and sinkage percentage value is given, but we are under null hypothesis. We are assuming that there is not any relations between two parameters, between biomarker and sinkage. And based upon these null hypothesis, we are calculating and we are getting the Spearman is 0.95. It is near to 1. And then we are saying that they are significantly related. Okay, so we are discarding our null hypothesis that they were not related. So that's why p-value is coming here as a point below 0.05. And definitely we can say that these two parameters are related with each other. Any points, any comments? Okay, let's say go for, I think we are almost touch. And final one is having Cohen-Mental-Hensel response adjusted for strata. So when we are having 2x2 response table across strata, we can go for adjusted odds ratio, that categorical way to control confounding without a full regression model. And it's not very oftenly used methods over there. I would prefer not to widely use it here. It is very, very rarely used in specific scenario. So can't I adjust pools every center response table 2x2? It is risky. If centers differ in case mix, pool table can reverse the directions, and then Simpson's paradox occur. So CMH computes the odds ratio within each strata, then pull them into one adjusted common odds ratio over there. So like here, MH stand with this responder in group 1 and center 1 and center 2, and responder in center B for center 1 and center 2, and then we are going to have the pool odds ratio. So once you are having data split with different strata, then we are going to combine it, but not roughly combination. So we are going to consider this, that representation of the centers over there, and thereafter we are going to calculate odds ratio. That's the beauty of having this. And again, let's say about this example with dashboard. So this is example with centered arm and arm A and arm B wise responders are given as how many were having yes and no. For center 1, for arm A, responder were 8, no non-responder were 2. Arm B were having 5 and 5. Arm A were having 3 and 17, 2 and 18, and this is where sorry, this is where we are having the pool odds ratio calculation. So point is that if you want to give this impact of the centers over there, definitely we can calculate this way. Otherwise, you can avoid it. And this is final one, but I would like to end it here. Its dashboard is there. We will calculate points on that. And yeah, any other point is there? Happy to have discussed with there now.
So the PSN would be used in as a non-parametric test to see the correlation between continuous values, right?
I couldn't hear. Could you repeat again? Sorry.
Spearman, what we saw was for continuous data, and when it is non-normal, as we saw in last time.
Yes. Let's open for discussion. Any points anybody is having? Do you have any chat questions?
Let me check. Does this substantial result means agreement from both of the test, or it simply signifies the chance?
Okay. So, does the substantial results mean the agreement from both of the tests, or simply okay? So, I couldn't understand what does it mean. Substantial results means. Chitrangshu, would you like to explain? Open your audio and try to explain it. Meanwhile, you are trying to, because I does substantial result. What's the substantial result you are saying? Okay.
I guess the substantial result would mean like any kind of kappa result with 0.8 or 0.9 means the agreement of both of the test, or it simply signifies the chance.
Okay. Okay.
Or the p-value.
Okay, I see. Yeah. I think the p-value ultimately we would emphasize on the p-value, and we will say it signifies the results. Okay, it's regarding Cohen kappa value. Yeah. Okay. So if your p-value below 0.05, then definitely we are going to say they are completely different, and we are going to discard that there is not any agreement over there. But if your p-value is above 0.05, that is a substantial results, test results, then we are going to say both raters are agreeing with each other. There is not any disparity between them. It's absolutely fine. Okay. Can quantitative exposure response model integrating toxicating exposure biomarker, molecular responder, and epidemiological data can be developed to estimate individualized disease risks analogous to pharmacodynamic model using drug evolution? So, yes. So, can quantitative exposure response model I think you are talking about much more the personalized risk determination model. Definitely, pharmacodynamics model can be used, and that's where the pharmacodynamics model used as a risk predictor, disease risk calculators. I would like to, Karthikeyan, we would like to get much more details on that. What the data you would be having, how you want to participate with these models to get the individual risk determinations. Please write to us with this registration link or email us. Definitely we'll be come back to you. But in principle, if I want to say, that's possible to have it. And drug evaluations been done by this manner, absolutely possible with the data that can be done. Najakat Ali says the feedback. Najakat Ali, so definitely, I think Ankush, we are having the email ID to connect, right? Have you have the registration link? Yes. Yeah, could you write it here in the message? So if you want to give some, or Najakat Ali, you want to get some specific template to having the feedback, but it's an open level, so you can write it, and it would definitely help us to improvise this, forthcoming one over there. Karthik, and you are saying that it's a much more the toxic dynamic evolution. Yeah, that can be done. Yeah. Karthik, and I am giving you overall superficial output, but I would like to get much more details about the scenario, about the data structure you are having based upon that. But apparently, but by looking your points, I'd say yeah, it's in principle it is possible to have it. Okay, dashboard, you can just link. Let me give you this link of the dashboard, and then definitely let me stop sharing, and dashboard is here. Just copy this dashboard at your place, and definitely you can get it, use it. Okay, and yeah, hopefully, yeah, dashboard link is there. And Ankush, if you can write this, or you can .coin. This is one email ID, and this is another email ID, and to share your feedback if you want to have it. Okay.
Yes,
Any
sir.
more points? Anything more?
So not from my side. Ankush, are there any questions from your side? Anything? No, sir, I guess. Yeah. So we have till now covered all the categorical testing, and sir has made it easy using the dashboard. Uh, I would suggest everyone to use the dashboard once, because showing is one thing and using is another thing. So the PPT has the examples and the dashboard has already upload in the dashboard. There is already uploaded these example data sets. So one has to just go and run and create such data as shown in the example and upload and run. It will definitely run and give you more feasible answers, and your understanding would be more clearer when you run it. We are happy to have any questions in next sessions or in emails or anything if you have regarding these sessions. And we suggest like let's go to the dashboard also and go to the PPT and run it once, and we can get back on this, and we can join for this another session as well further.
Pawan, please feel free to use this dashboard, and for you guys only, we prepared this dashboard.
Yeah.
Use it.
If there are any another concepts that you want to have light on, you can suggest here as well, or on mails, or any feedback or registration forms, you can let us know. We will incorporate those things which are useful in your work, day-to-day work. Or if possible, if anyone is dealing with any kind of example data sets or anything work like can share some problem, we can discuss it if required. Yeah, for the last session as well, we have created a dashboard. It is in work in progress state. It will be also be deployed and can be shared to you guys. However, I would suggest you also be stick to SPSS as well, because anyways you guys will have to generate those SPSS data for your day-to-day life. You cannot skip those things being in TMH. So yeah, you have to be alert with SPSS, but we'll also share the dashboard for that as well for
Yeah,
testing.
I think we are having
Yeah.
that LinkedIn
Yeah.
page, and please feel free to join over there. So in upcoming time, we will definitely share other dashboard and other creations. So it will be easy for all use it over there. To be honest, or one of the goal is to come out from the proprietary software like SPSS. SPSS takes a huge cost, and many times once you come out from your medical institute, you are going to go independently, because many of you are studying and many of you will be having your own center. So it is very difficult to having this SPSS again to buy and purchase annual subscriptions. It's quite a lot. So our intention objective is to empower this analysis, to make it free. As much as it's not free does mean it's a we are doing rigorous research work to make it much more effective. And by today's lookout, you have seen that how easy or computationally rigorous things being done in background to get it done. So we are trying to empower these others to use this, enrich the medical research. So it will be completely free in future as well. So please feel to use it.
Yeah.
Sir,
Thank you, sir.
somebody is asking also about the program schedule. If we can share the topic, next we are going to discuss.
Okay, share the
Schedule.
program schedule. So here, to be honest, we are not sticking with the schedule program. We are trying to make, by responder, we are doing different data analysis here, by how many were, because once we are registering, we are looking your your presence here. We are trying to do any in-depth analysis, because as a statistician we do, and based upon that we are next time what the program title would be, that we are deciding. But one thing we can say, that we are trying to touch the basic platform over there, that's very rarely been touched upon. We are not going to have very advanced one until recently. So we will go slowly over there. So our objective is to, again, we long time we are investing in this academic research. Our objective is to empower this analysis program much more better, that you guys use in your routine life, in your master dissertation, your PhD dissertation, much more better way. So based upon that requirement, we will try to figure it out what else or what topic could be touched over there. But definitely we will touch your real requirement that can serve your purpose. So that be assured that we are not going to give any topic that is not going to be used in your So broadly, whatever the topic we are going to touch, by and large, it is going to be used for your upcoming work somehow. Be assured that. But specific title we are not going to announce now. We will be announcing with our flyer, with our upcoming flyer, once we're going to announce in next time.
Sir, I have one request, sir. There are many clinical studies scattered over the internet, but can we have a single review paper? We can find the all biostatistical analysis, statistical analysis. As a beginner, I want to learn the clinical things, statistical analysis over the biostatistical data. So, could you please suggest one review paper for us?
So basically you want to get some guidance or guideline out of that, is it?
Yeah, yes.
Okay. So okay, that's a very good point. I can could you share your email ID here?
Yeah, definitely,
You
sir.
don't
In the
mind?
chat box, I am ready.
Yeah, please share some of your email ID. Then we can send to you that guideline or articles over there. So, but by
Yes.
the could you say that exactly what you are looking? What is your background? So we can give you some better
Sir,
specific
in the
one.
domain of clinical disease, whatever the biostatistical analysis we
What
do
clinical trials you are? What
is
specific?
not specific for the cancer disease, sir, any kind of disease. Because I want to switch into the biostatistics domain. So, as a
Okay,
beginner, I want to learn
I
something,
see.
what kind of
Okay,
researches are going.
okay. So
So,
you are
in a
not.
single, please.
So you're not stick with any specific therapeutic domain. You are open with anyone. Okay, that's what. Because I was not aware about your background, so that's the thing.
Sure.
Perfect. Okay. Perfect. Yeah, recording would be there. Can you get the recording? Again, use our LinkedIn profile page, and also that our profile page, where you will also get our YouTube link, where entire tutorials been recorded and uploaded. Ankush, if correct me if I'm wrong. Also, we are having this TMH TMC website. We are also uploading with the link, or we are uploading. How we are uploading that part?
Yes, sir, we shared in mail also, and our site also there is recording sessions.
So definitely all the lists are available. All the links are available with the tutorial. in in YouTube. Perfect. I mean, we're really excited about after having your all participants, and we will go ahead with this. Perfect.
So I guess we can close for now today.
Yeah, we can close it. Yeah, sure.
So thank you all for connecting. I hope the statistics knowledge helps you in your clinical research, and that is what the motto is. We look forward to connecting for more sessions. We'll bring more interesting sessions. Do let us know what you want to learn and more about the statistics for your domain, particular domain. And that is very important for us to conduct next session as well. We'll let you know the topic and this through mails. And thank you for connecting. Thank you. Have a
Thank
nice
you.
day.
And Ankush, could you retrieve this email ID being listed in this chat box now? Once you close this session,
Yes, sir.
or you
Yes,
need
sir.
to copy it now itself?
Sir, I'm sharing. I retrieved. I shared in.
In your
I've saved it. Yeah.
we'll be having this video, but still, it is if you can copy it now.
Sir, I've saved it. Yes.
Perfect.
Okay.
Okay.
Bye.
See you guys then. Okay.
Thank you.
Bye then.